Hello friends and welcome. Today we shall talk about water testing. In today's deliberation, we shall try to understand the importance of water in food processing industry. Also, we shall try to describe the sources and quality of water, followed by describing various tests that are used to assess whether the water is potable or not. Water, like food, is a potential vehicle for the direct transmission of disease-causing agents. It has been and continues to be a reason of significant outbreaks of disease in developed and developing countries. Water quality and food safety are actually inextricably linked. For example, drinking water was identified as the source of a significant and fatal outbreak of Escherichia coli in Canada, as reported in a study by Condro in 2000. In 1970, a cholera epidemic in Jerusalem was traced back to the consumption of salad vegetables irrigated with raw wastewater, and in 2006, a chocolate company located in the United Kingdom was affected by contamination of chocolate by salmonella from a leaking wastewater pipe in the production area. Water is therefore capable of introducing contamination into food if appropriate care is not taken. Although water is just one route by which foods can be contaminated with disease-causing agents or toxic chemicals, it is potentially very important. Raw water can be contaminated with pathogens, usually as a consequence of human or animal fecal matter, or a wide range of natural or anthropogenic chemicals. Contamination can arise in water as a consequence of improper storage, pickup of contaminants from distribution systems, or leakage from a dirty water system into a clean water system. In this way, water may present some risks to food safety as it is an essential part of food production and processing. It is therefore vital to minimize the unnecessary risks associated with the use of contaminated water, which in turn necessitates the water testing operations. In this module, the importance of water testing and the different assays recommended for this purpose will be discussed. Let's firstly discuss the importance of water in food processing industry and the source of its contamination. Water is important to the food processing industry because it is not only present in all foods but is also extensively used in most food plants as a processing aid and for cleaning operations. When water is used as a food ingredient, its quality, for example impurities, can affect the properties of foods including texture, shelf stability, appearance, aroma, and flavor. As a processing aid, water may be used for conveying, heating, cooling, rinsing, dissolving, dispersing, blanketing, diluting, separating, steam generation, and other activities. In each case, purity of the water will affect its performance. Critical agents that deteriorate the water quality when talking about food industry can be distinguished in microbiological contaminants, chemical contaminants, and physical contaminants. The risks associated with the use of water contaminated by one or more of such agents in different foods include: firstly, the presence of the pathogens of fecal or non-fecal origin that cause foodborne illnesses. It may in turn include bacterial pathogens like Vibrio cholera, Salmonella typhi, and pathogenic strains of Escherichia coli. Viruses, especially enteroviruses, protozoa, especially Giardia species, Cryptosporidium species, and Ant amoeba histolytica. and algae that are chiefly responsible for the eutrophication. Secondly, the presence of harmful chemicals, and thirdly, the presence of chemicals that are not harmful but have a concentration above their allowable daily intake, that's ADI. In order to minimize such risks, an approach of formalizing quality assurance programs for drinking water supplies has recently emerged, where testing the quality of water plays main role for hazard identification and for taking the corrective measures. Let's now discuss some water quality requirements in food industry. Food industry has a high demand for clean and fresh water. Access to an abundant source of high quality water is one of the essential factors in designing a food plant. In general, the uses of water in different sectors of food industry can be classified into two main categories, depending on whether the water comes into contact with food products or not. The examples where water has no direct contact with the foods include the use of heating or cooling and for generation of non-food steam. On the other hand, the instances where water has contact with foods may be at the raw product stage, for example washing or transport, at intermediate stages, for example cleaning of equipment, or in the final product itself, for example when water is used as an ingredient. Precisely, quality parameters of water for the different production systems can be discussed under 4 subheadings, which describe its most intended applications. These include the uses of water as a food ingredient, as bottled water for washing and rinsing, for blanching, and for culinary steam. When water is often a direct ingredient in food and drink, its quality can have significant impact on the safety, quality, and taste of products. It must be free from undesirable taste, odor, color, and impurities that could be harmful to consumers and product quality. In addition, it must be free from physical, chemical, and microbiological hazards. Raw water can be contaminated with pathogens, usually as a consequence of human or animal fecal material or runoff contaminated with fecal material. It can also be contaminated with a wide range of chemicals, both natural and anthropogenic, which are of concern under some circumstances, depending on the concentrations present. The chemical and microbiological hazards in water generally arise from the disposal of untreated sewage containing high microbial load and toxic chemical residues in the water bodies by various industries and the agricultural sector. On the other hand, if we are talking about the bottled water, it is generally required to be bacteria free. In order to achieve this, many bottled water processors use ozonation, ultraviolet treatment, and or reverse osmosis, which removes 99.9% of all viruses, bacteria, and pyrogens, and is more energy efficient compared to others. For applications of washing and rinsing, water used must be clean and soft. Mild taste and odor issues are not much important, but its hardness should be removed along with the mineral content that could affect the additive performance. Water used for blanching is required to be free from thermophilic bacteria. It may, however, contain some added chemicals like sodium carbonate or calcium oxide up to a certain concentration to protect chlorophyll and to retain the color of green vegetables. In contrast, the water used for generation of culinary steam, that is in general directly injected into a product, may be one of the most difficult treatment cases in the food industry. It requires to be thoroughly treated to prevent problems in boilers and associated piping, valves, and processing equipment. Corrosive components like oxygen and carbon dioxide may be removed by deaeration. In addition, the pH adjustment is essential for this application. Boiler water pH should be maintained at about 8.5 and may require the addition of chemicals for this purpose. In this regard, food producers, processors, and caterers need to ensure that the use of water in their premises is maintained to the highest possible standards. It's therefore vital to minimize the risks and avoid unnecessary harm by ensuring that the necessary control measures are put in place. For example, water safety plans and hazard analysis and critical control point called HACCP in order to ensure the safety and quality standards of water for the specific applications. Its testing is highly essential for every sector and subsector of food industry. This brings us to the most vital part of this discussion, that is different types of tests conducted for testing the water quality. We'll explain the different methods used for testing the quality of water one by one. The first section of tests used to determine water quality belongs to the bacteriological analysis. The principal risk associated with water is that of infectious diseases related to fecal contamination. Hence, the microbiological examination of drinking water emphasizes the assessment of hygienic quality of the supply. This requires the isolation and enumeration of organisms that indicate the presence of fecal contamination. In certain circumstances, the same indicator organisms may also be used to assess the efficiency of drinking water treatment plants, which is an important element of quality control. Commonly used methods for bacteriological analysis of water include multiple tube method, membrane filtration method, presence-absence test, and single application or disposable test kits. Firstly, we shall discuss membrane filtration method. In this method, a minimum volume of 10 milliliters of the sample is introduced aseptically into a sterile or properly disinfected filtration assembly containing a sterile membrane filter with a nominal pore size of 0.2 to 0.45 millimeters. A vacuum is applied and the sample is drawn through the membrane filter. All indicator organisms are retained on or within the filter, which is then transferred to a suitable selective culture medium in a petri dish. Following a period of resuscitation, during which the bacteria become acclimatized to the new conditions, the petri dish is transferred to an incubator at the appropriate selective temperature, where it is incubated for a suitable time to allow the replication of the indicator organisms. Visually identifiable colonies are formed and counted, and the results are expressed in numbers of colony forming units called CFU per 10 milliliters of original sample. This technique is inappropriate for waters with a level of turbidity that would cause the filter to become blocked before an adequate volume of water had passed through. Secondly, we shall discuss the multiple tube method. Multiple tube method depends upon the separate analysis of a number of volumes of the sample. Each volume is mixed with culture medium and incubated. The concentration of microorganisms in original sample is then estimated from the pattern of positive results, that is, the number of tubes showing growth in each volume series by means of statistical tables that give the most probable number per 100 milliliters of the original sample. Therefore, it's also referred to as the most probable number or MPN. Thirdly, we shall discuss the presence-absence tests. Presence-absence tests are qualitative tests that indicate only the presence or absence of the indicator microorganism sought. The use of such tests is appropriate only where positive results are known to be rare, as these are not meant to quantify the degree of contamination. Consequently, presence-absence tests have a very little use, particularly in countries or situations where contamination is common, and the purpose of analysis is to determine the extent of contamination rather than whether or not the contaminant is present. These are not recommended for use in the analysis of surface waters, untreated small community supplies, or large water supplies that may experience occasional operational and maintenance difficulties. Fourthly, we shall discuss single application or disposable test kits. Disposable test kits provide the results of microbial contamination very rapidly. These are both widely marketed and increasingly used in developed countries. However, their reliability may vary widely and should be properly assessed by a reference laboratory. Further, an important assay in microbiological quality assessment of water is heterotrophic plate count, formerly known as standard plate count or total viable count. It's a technique used for estimating the viable heterotrophic bacterial population in a sample of water. The general principle of this method is that a sample of water is put on the Petri plate that contains nutrient media required by bacteria for growth and survival. The nutrient media that is most often used for this test is called R2A agar, which is a gelatin-like substance best suited to the needs of water bacteria. After 5 to 7 days of incubation, the number of small spots on the plate called colonies is counted, and a measure of how many bacteria are present in each milliliter of water is determined. The HPC results are generally reported as colony forming units per milliliter or CFU per ml. Each colony forming unit represents an initial single live bacterium that was capable of multiplying until it could be observed on the plate. It's important to understand that the colony count alone does not allow one to draw conclusions about the risks to public health. However, it currently serves as a relatively easy way to measure the filtration and disinfection efficiency as well as the estimated number of bacteria in areas that have the potential for increased contamination. After microbiological tests to assess the water quality, the second category belongs to physicochemical analysis, which includes chlorine residual, pH determination, and turbidity. Chlorine is the principal disinfecting agent used for water treatment in most countries due to the advantages like relative cheapness, efficacy, and ease of measurement, both in laboratories and in the field. However, the presence of free chlorine, which forms the most reactive species like hypochlorous acid and hypochlorite ion in water, must be monitored. The method recommended for the analysis of chlorine residues in drinking water employs NN-diethyl p-phenylene diamine, more commonly referred to as DPD. The addition of DPD to the water causes the generation of color, which can be measured spectrophotometrically. Most commonly, color generated is matched against the standard colored discs or tubes. The method can be used by staff without extensive specialized training. In addition, analysis can also be made using starch potassium iodide, which is not specific for free chlorine but measures directly the total of free and combined chlorine. It's therefore not recommended except in countries where it is impossible to obtain or prepare DPD. In addition to chlorine residual, it's important to measure the pH of water since the efficacy of disinfection with chlorine is highly pH dependent. In case the pH of the treated water exceeds 8.0, disinfection is less effective. To check that the pH is in the optimal range for disinfection with chlorine, that is less than 8.0, simple tests may be conducted in the field using comparators such as those used for chlorine residues. With some chlorine comparators, it is possible to measure pH and residual chlorine simultaneously. Alternatively, portable pH electrodes and pH meters are now available that provide the reproducible and precise information about the pH of the sample. However, if these are used in the laboratory, they must be calibrated against fresh pH standards at least daily. Third important physico-chemical property to be determined in potable water is turbidity. High levels of turbidity can protect microorganisms from the effects of disinfection, stimulate the growth of bacteria, and exert a significant chlorine demand. Where disinfection of water is practiced, the turbidity must always be low, for example below 5 nephelometric turbidity units, that is NTU, or more specifically below 1 NTU for effective disinfection. Measurement of turbidities lower than 5 NTU will generally require the electronic meters, while those above 5 NTU can be measured by simple extinction methods, which are far cheaper and require no consumables. In the monitoring of small community supplies in developing countries, such methods may be more preferable. Fourth essential physico-chemical test required for water testing is alkalinity. Alkalinity is a chemical measurement of the ability of water to neutralize acids. In natural waters, alkalinity is due to the presence of weak acid salts, carbonate and bicarbonate species. To determine the alkalinity, a known volume of water sample is titrated with a standard solution of strong acid to a pH value in the approximate range of 4 to 5. Titrations can distinguish between three types of alkalinity, that is carbonate, bicarbonate and total alkalinity. Carbonate alkalinity is determined by titration of the water sample to the phenolphthalein indicator endpoint, or approximately a pH of 8.3. Total alkalinity is determined by titration of water sample to the endpoint of methyl ORIG indicator, or an approximate pH of 4.5. The difference between the two is bicarbonate alkalinity. Finally, the last category of tests conducted on water include the determination of aesthetic parameters. Aesthetic parameters of water include color, taste and odor. They are important in monitoring community water supplies because they may cause the water supply to be rejected. Color in drinking water may be due to the presence of colored organic matter. For example, humic substances, metals such as iron and manganese, or highly colored industrial wastes. Drinking water should be colorless. For the purpose of surveillance of community water supplies, it's useful simply to note the presence or absence of observable color at the time of sampling. Changes in the color of water and the appearance of new colors serve as indicators that further investigation is needed. Other two aesthetic parameters, that is taste and odor, are also important in determination of aesthetic quality of water. Generally, the taste buds in the oral cavity detect the inorganic compounds or metals such as magnesium, calcium sodium, copper, iron and zinc. Water should be free of objectionable taste and odor and should not be offensive to the majority of the consumers. If the sampling officer has a reason to suspect the presence of harmful contaminants in the supply, it is advisable to avoid direct tasting and swallowing of the water. Under these circumstances, a sample should be taken for investigation to a central laboratory. It's important to mention that there are some other tests also that are conducted on water and have relevance to health of consumers. Some of these health-related parameters include fluorides, nitrates, lead, chromium, arsenic and pesticides. If these or any other chemicals of health significance are thought to be present, they should be monitored and examined in the light of World Health Organization guideline values or any relevant national standards already established. These parameters may be measured by means of portable test kits based on conventional titrations, comparators or photometers. If this is done, the reagents must be of high quality and carefully standardized. Other parameters require conventional laboratory analysis by spectrophotometry, atomic absorption spectroscopy or chromatography using standard methods. With this, we come to the end of today's lecture. I hope you have understood it well. Thank you very much and goodbye.